Hey everyone, this is Lad from Astrolabe Diagnostics, and today I'd like to talk about the latest feature that we're releasing for Astrolabe, and that is a complete redesign of the Cell Subset Navigator. When dealing with high complexity flow, there is a lot of information to deal with. There's many different cell subsets, there's many different covariates and research questions that you want to ask. The Cell Subset Navigator is the tool that takes all of that information, all of these hypotheses, all of these ideas, and puts them in one place so you can readily and effectively scroll between them and pick the ones that are most relevant, are most interesting, and really promote your research the most. And this is how the navigator looks like. This is a composite view of the entire experiment. So everything I'm showing you here is using data from all of your FCS files, whether you have 5 or 50 or 500. And the first thing we see here is a list of the different cell subsets that Astrolabe identifies for you. Astrolabe does this automatically. This is dependent on your panel. So the platform reads your FCS file, sees which markers you use, and decides which subsets to look for. For you as a researcher, this means no cleaning, no manual gating, no doing your own statistics. The platform takes care of all of these for you in just a couple of hours. You upload your FCS files, you come back in 2 hours, and you can start playing with the data. So, let's say that we want to look at a specific population, for example, the effector memory CD4 T cells. We can just click on it and scroll down, and now we're going to see the frequency of this population in each one of the donors in this study. So again, all of the FCS files are here. Now, this is a good time to mention that this is an immune monitoring study that compares two populations, American children from Stanford, California, and Bangladeshi children. So let's overlay that information on top of the plot. I'm going to click here and choose source. And now, we're seeing a box plot with aggregate statistics comparing the two populations, and all of the single sample data is color-coded by the population. And we can see that for the effector memory CD4 T cells, there isn't much of a difference. The median is 1.5% for the Bangladeshi group and 1.1% for the American group. However, we can now slice this according to other covariates. For example, we can look at ages, different ages. So now we're comparing the 1-year-old kids, and you see how the interface immediately adapts. We're looking just at the samples for these children. Still not much of a difference. Let's switch to the 2-year-olds. And now we can finally see there's a difference between the groups. For the Bangladeshi, it's about 2.3%, for the American, 1.1%. So more than a twofold change, and this is also reflected in the sample by sample or donor by donor data. And finally, we can switch to the 3-year-old kids, and once again we see a twofold change. There's a bit more variability in the American population, but the values for the Bangladeshi group is much higher. So, as you can see here, I can easily flip between different hypotheses, different ideas. For example, let's say I just want to look at the Bangladeshi children and compare the different ages. The interface immediately adapts, and we can see the different donors, color-coded by age, and we can see the box plot, and we can see how, as this population matures, the frequency of these cells go up, compared to the American population, where there is an increase, it's just not as deep as the Bangladeshi group. One last thing I want to mention is that if you don't know which cell subset you want to look at, we have this handy volcano plot visualization to point you out at those cell subsets that are the most significant. So we see the effector memory CD4 T cells, and they seem to go up as the American group matures. At the other side of the volcano plot, we're going to see the central memory CD4 T cells, and I can click on them and scroll down, and now we can see the trend for the central memory CD4 T cells. They start highest at the 1-year-olds, then go down, and then go up again. If I want to look at this trend in the Bangladeshi group, I'm just going to flip the Bangladeshi. As easy as that. So we can see that there's an increase in the effector memory, but the trend for the central memory is not as clear. And finally, if you want to compare the 3-year-old kids, it looks like the population of these cells goes down from the Bangladeshi to the American for the effector memory, but there's a slight increase for the central memory. So once again, the idea is to navigate through different hypotheses, through different ideas, very easily, very quickly. In order to reach here, you don't need to do anything. You just upload your FCS files, and that's it. So Astrolabe is really built around allowing you to access your data and unlock the ideas, the insights, the intelligence as quickly and effectively as possible. If you need help analyzing your set of data, please reach out. I'm Elad at astrolabediagnostics.com, and I'm looking forward to talking about your experiment and see how Astrolabe can help.